Still on point.
No, it's off.
Okay. Okay, well, thank you, Carlos, for the invitation. It's great being here in Concepcion again. I always like coming here. It's nice and green. The campus you have here is fantastic. And also the company. I'll be talking today about one of my favorite topics, and that is the role of caveolin in cancer, and specifically how it contributes to metastatic disease in exosomes. So where do I point this? Over there? So just a little bit about our lab. We are obviously located in Santiago, as Carlos pointed out. Whoops. And we're part of two centers, basically this Advanced Center for Chronic Diseases and this center I actually direct which is a Center for Studies on Exercise, Metabolism and Cancer. And basically in these two centers, our focus is cancer, but on different aspects. So this is the lab, and essentially we're two groups. A group of Lisette Layton, my wife. She works on these really nice cells here, astrocytes, how they interact with neurons, and basically the process of astrogliosis and how that inhibits neuronal regeneration. And in my part of the lab, we focus on these somewhat more ugly cells, cancer cells, and we study signaling mechanisms that either contribute to tumor suppression or, in the particular case today, promote metastasis. Just like to mention the people involved in the studies I will talk about today. So we have a group of young investigators we collaborate with. These are all people who trained at some stage in the lab and now have independent positions. So Vicente Torres, Lorena Lobos, Rino Ortiz, and then we have postdocs. They'd like to highlight Jorge Diaz and Natalia Diaz, who just left the lab. And amongst the graduate students, America Campos. She was instrumental in setting up everything that has to do with exosomes in the lab. And then these people in blue are the rest of the exosome group. So Ricardo, Renato, Maria Fernando, and Pablo Lara. So I didn't quite realize when I put this talk together that I was at the interface between gestational diabetes and cancer. So I didn't put a lot of introductory slides in. But basically in the lab in my part of the lab, we work on three main topics. One is related to cell death mechanisms. Also more recently, inflammation has become of interest. We look at things like bacteria-cell interactions and how this induces signaling events in cells. Particularly, we're looking there at Helicobacter pylori and its role in gastric cancer. And then we have basically a research area focusing on this protein, caveolin-1, and how it contributes to the development of progression of cancer. And so this is a rather low-resolution image of the caveolin structure. There is no really good structure. It's a membrane protein, so it's very difficult to get. Plus, it oligomerizes, as you can see here. And so basically we have a central hydrophobic core region here, and then we have these two cytoplasmic tails. This one associates with the membrane. This one sort of sticks out of the membrane and can suffer then modifications like this phosphorylation on tyrosine 14 here, which will become important. This protein has the ability then to dimerize and then oligomerize, as you can see here. And these oligomers are generated in the trans-Golgi network. Basically, here the protein associates with cholesterol sphingolipid-rich microdomains. They're also referred to as rafts, present in the trans-Golgi. And then, as such, go to the plasma membrane and form there what are called planar caveolae. Then other proteins associate with the caveolin to generate these structures here that are what you can see in a microscope, an electron microscope, these invaginations of the membrane that are called caveolae. Now, by definition, these structures are, of course, involved in transport, vesicular transport, and obviously caveolin plays an important role there. Caveolin is also important in things like cholesterol homeostasis. because it binds cholesterol, and it's important for instance, taking cholesterol from the ER to the plasma membrane. The aspect we're interested in, since we're a signaling lab, is its role in signal transduction. And so in the 90s, when I sort of got interested in this protein, basically what they were publishing was that it was a protein that could interact with a large number of proteins involved in signal transduction, some of them are shown here, and inhibit their activity. Many of these are sort of oncogenic, and so by definition, it was determined it was a tumor suppressor. I thought that was an interesting problem to look at. And our entree into the field came with a publication where we showed actually in colon cancer cells that it could function as a tumor suppressor and that it was downregulated in colon cancer tissues at the mRNA level. But then we also had a sort of evidence looking at some cell lines that had a greater metastatic potential, that in those cells the caveolin levels increased. So it seems like initially in the transformation process it's lost, and then it can be upregulated again. And so this put together in a number of reviews throughout the years. We tried to summarize data available in the literature and came up with this kind of model. And what you're looking at here is the relative caveolin expression levels in tissues. So breast, lung, colon, red, prostate, green. And so there are like two classes. Tissues that express caveolin and tissues that don't. And in the tissues that express caveolin, what we frequently see in early phases of cell transformation is the loss of expression. So this is a epigenetic phenomenon, methylation of the promoter region and silencing of the gene. Then there's a long interphase where nothing much seems to happen. And then when we get to later stages of tumor development, when we go into, for instance, basically the metastatic phase, we have an increase in expression again. And the increased expression there is associated with poor patient prognosis, for instance, certain types of cancer like prostate cancer. And if we look at different types of cancer cells, advanced cancer cells that express caveolin, it will promote in vitro migration, evasion, and metastasis. So basically what we've been doing in the last 15 years is trying to figure out how you can transit from here, tumor suppressor, to here, promoter and metastasis. Well, this is basically the summary. And so essentially, I often refer to caveolin as a Jekyll and Hyde in cancer. So Jekyll being good, Dr. Jekyll, Mr. Hyde being bad. So this is caveolin, the good one, acting as a tumor suppressor. We showed that it's present in a complex together with E-cadherin, where it enhances sequestration of this protein here called beta-catenin. And as probably many of you know, beta-catenin can translocate to the nucleus and promote transcription of genes, depending on this family of transcription factors called the TCF-LEF family, of genes like cyclin D1, cyclooxygenase 2, and survivin. So this would be cell cycle control, inflammation, cell survival. Obviously, all of this is important to cancer cells. So this complex would prevent beta-catenin from doing this. So would reduce all of the expression of all of these genes. In doing so, increase cell death, reduce proliferation. So this is how we explain at least in part, the tumor suppressor effect of caveolin-1. In addition, the presence of E-cadherin, basically in the presence we have suppression of metastasis. So this is well known. Caveolin on its own transits then to this form here, just shown as a dimer, not as an oligomer. We don't really know which state the protein is in. But what we do know is that in the absence of E-cadherin, caveolin there can promote a signaling cascade that leads to metastasis. And I'll show you how. Before that, I just want to mention one point that I think is highly relevant here since we've been talking about inflammation. What we've also shown, I won't go into detail, is that this complex is not immune to inflammatory processes. So we know that inflammation is a contributing factor to the development of cancer. And what we've shown in a series of experiments is if you treat cells expressing caveolin E-cadherin, which basically will not form tumors, in an animal model. If you pre-treat the cells with pro-inflammatory cytokines, you can show that this complex dissociates and then in a covalent-dependent manner promotes metastasis. So this is in vivo. So that means in a situation where you have a high level of inflammation, this tumor suppressor complex stops to function as such. So this helps us in understanding how, amongst other things, that inflammation can contribute to the development of cancer. So I'm just going to focus now, as I said, on this part of the curve where we're looking at caveolin-1 and how caveolin-1 promotes migration, invasion, and metastasis. And we're going to start off by looking at cell intrinsic mechanisms, so what happens just within the cell. And then in the second part, I'll say a little bit about how we think exosomes might be involved. So just to show you how we came up with our signaling scheme downstream of caveolin, a few simple experiments. One thing you can do with cells is, of course, let them sit down on a surface, look at how they spread, and then how they start migrating. And the nice thing about this very simple experiment is that you can look at how the cell interacts with different matrices. So you can, for instance, look at fibronectin or vitronectin or collagen and then see whether all surfaces behave the same way. And the experiment is rather simple. You just look at the morphology of the cells and how they increase in area and then eventually start migrating. So you get a plot here of area over time, and it looks something like this. If you do this experiment now, for instance, looking at spreading on fibronectin, these B16F10 cells, so this is a melanoma cell line where we've introduced caveolin. And what you can see is that the phosphorylation of caveolin increases rapidly as the cells basically adhere to the surface. So this was here. Then it goes down again, and as they start migrating, basically we have an increase in phosphorylation of caveolin here, as evidenced by an antibody. And we have an increase in size here, and then they're clearly going into a sort of a more migratory phenotype, or developing a more migratory phenotype here between 45 and 60 minutes, associated with this increase in migration and phosphorylation. So this is basically an experiment that relates or correlates phosphorylation of caveolin with enhanced migratory phenotype. Then you can do the same experiment on a matrix like vitronectin, and essentially what you see there is, first of all, the cells clearly do not seem to like very much this matrix, and also if we look at phosphorylation, there's a minor increase here, and basically here at 60 minutes we almost don't see any increase whatsoever. So this is they do migrate eventually, but they do so more slowly in a less directional manner. So what caveolin does in these cells in a phosphorylation-dependent manner is favor migration and actually favors directional migration. So they're faster and they're more directed. Now, when thinking about migration, invasion, or even metastasis, the proteins that come to mind are these small g proteins like CDC42, RAC1, or RhoA. All of these are implicated in different aspects of the migration process, so either filopodia formation, lamellopodia formation, or focal adhesion formation. And so we basically wanted to know downstream of caveolin-1 which of these proteins might be activated. And a simple way to do this, relatively simple, is doing what we call pull-down assays. So basically what you do is you try and identify the activated form of the small g proteins. When small g proteins are activated, essentially they bind GTP. That's their activated state. And so you basically have a bait protein which will only bind specifically to the GTP-bound form of whatever G protein you're interested in. And the bait will vary depending on the G protein you want to pull down. So we did this with the different G proteins. And basically what we found is shown here. So this is now an aggressive breast cancer cell line called MDA-MB231, which has high endogenous levels of caveolin. And what you can do is, of course, knock down the caveolin in these cells. And you do a control experiment, which basically, of course, doesn't knock down the caveolin levels, but the cells go through the whole transduction process. And this would be the control cells, and these would be the knockdown cells. And if you do this pull-down assay for RAC1, what you see is in the covalent-expressing cells, as the cells spread and then start migrating here, remember they first sort of sit down, spread, and then start migrating, 60-minute time window here, you have an increase in RAC1 GTP loading. So that means activation of RAC1, not so in the cells where you've knocked down covalent. So this means you have covalent and somewhere downstream RAC1 associated with lamellipodia formation, important for migration. Okay, we did this with the other proteins, basically like row A. We also get an increase, but there is no difference between cells with and without covalent. Here's another experiment now using melanoma cells, human melanoma cells, and here too, we increase covalent expression that coincides with higher levels of RAC GTP loading, so higher RAC GTP activity. So we basically have covalent now and RAC1. How do we connect them? We were inspired by this publication here, which showed that in certain circumstances, Rab5 can act as an activator of RAC1. Now, Rab5 is, again, a small G protein implicated in endocytosis. And so what they showed there is that Rab5 activates a guanine nucleotide exchange factor called TM1, which activates RAC1, and that's important for migration. So at the time, Vicente Dores had just got back from his postdoc, and he was really interested in Rab5, and so we said, well, let's give it a shot and see whether Rab5 is involved. And so, of course, Rab5 is involved. Let's just focus on this experiment here, which is a transmigration assay. We're looking at colon cancer cells and melanoma cells, colon cancer, melanoma, and we can introduce covalent into these cells. So these are the cells with a mock plasmid. These are the cells with a covalent-expressing plasmid. And what you can see is the migration of these cells, transmigration increases. In these melanoma cells, the same thing. And now, if we generate the, oh, what's this kind of on the loose here? Okay. And if we generate these Rab5 knockdown cells, am I doing something here? I don't know. Ghost in the machine. Anyway, so these are the Rab5 knockdown cells, the white bars, and essentially we do not get this covalent-dependent increase in migration, neither in the colon cancer cells nor in the melanoma cells. So that means that we did other experiments with RAC1 to show the same thing happens, and also we did experiments to show that actually Rab5 is between covalent-1 and RAC1. So we wanted to show that this is relevant in vivo too. So we use these, as I said, B16F10 melanoma cells that are derived from these B16F0 cells. And essentially what was done was they were injected into the tail vein of these mice and recovered from the lungs. And this was repeated 10 times, and that's how you get the B16F10 cells. So they have enhanced metastatic potential and specifically go to the lung. That's what you want in a metastasis assay, because if you have to go and look for metastasis all over the body of a mouse, it's rather difficult. So this is the assay. So we inject into the tail vein here, and then we score for metastasis to the lung. And this is what you see. So these are the lungs without fixation, here after fixing. This is lungs from mice where there was no metastasis, lungs from mice where there was metastasis. So it's really easy to identify the black tumor mass due to the melanomas in the lung. And if we now compare the cells without covalent to the cells with covalent, you can see there's about a 3-fold increase in metastasis to the lung. And if we do the appropriate knockdown experiment, for instance, with Rab5 here, so this is the experiment, the mock cells, covalent-1, we get an increase in metastasis to the lung. And if we knock down Rab5 in these cells, we basically abolish that. So it's true in vitro. We have this sequence, and this sequence is relevant for metastasis in vivo. So without showing all the data, basically what we came up with is this model in the lab. So covalent is in this complex with E-cadherin if the cells express E-cadherin. This is a tumor suppressor complex. And basically what happens in this complex is a phosphatase recruited, which I'm not showing here, a novel phosphatase called PTBN14, and it dephosphorylates covalent. So covalent cannot act as shown on the right here. In the absence of E-cadherin, it gets phosphorylated by these Sark family kinases, recruits an adapter called P85, which I didn't mention, and this at P85 is a gap for RAB5, so basically it inactivates RAB5. If we sequester the gap, RAB5 is activated. On the early endosomes activates TM1 and RAC1, which then goes to the plasma membrane and promotes migration, invasion, and metastasis. As I said, so this is the cell intrinsic part of the mechanism, and we sort of became interested in the possibility that caveolin also might be doing something in exosomes. There was a number of reasons to believe that that might be the case. For instance, in prostate cancer patients, it was known that caveolin in some form or other levels increased in plasma samples of patients that had advanced stage prostate cancer. And as already mentioned, in that particular cancer, it was also known that increased caveolin expression was related with poor prognosis, and this is also true, for instance, in melanoma, breast cancer, and so on. So, we're going to talk now a little bit about the power of caveolin-1 containing exosomes. So, essentially, this is the idea here. You have a cell here that can produce, of course, a number of different types of vesicles. We already heard about that from Carlos. Can produce microvesicles or exosomes in these multivesicular bodies, which confuse them with the membrane and release the content, in this particular case, the exosomes. For our experiments, we used this cell line I already mentioned, these MDA-MB231 cells, which is a highly metastatic breast cancer cell line, and these cells express high levels of caveolin. And the idea is that the transfer of exosomes to another cell of lower metastatic potential, and this was actually shown in the literature, to MCF cells, FCF7 cells, another breast cancer line. And what they showed is that transfer of exosomes from this cell to this cell increased migration invasion of these recipient cells. They did not link that to the presence of caveolin. But, of course, we know or knew that these cells have a low level of caveolin expression, and these other cells have a high level of caveolin expression. So the question, of course, was, may this transfer of this metastatic capacity somehow relate to the presence of caveolin in these exosomes? So this is sort of was the working model. We said, okay, the donor cells of higher metastatic potential would produce or should produce exosomes containing caveolin. These would then fuse with the recipient cell, and somehow the most likely scenario would be that the caveolin introduced into these cells would then promote signaling via this cascade and increase migration, invasion, and metastasis. Turns out the first part is true, and the second part is probably not true. Anyway, so talking about exosomes, of course, we now know that they contain a large number of different types of factors. So they have membrane proteins, soluble proteins inside. They also contain bits and pieces of RNA, microRNA. They also have a specific lipid composition, which people tend to not consider very much. They have a defined size. And so, of course, they also, based on their genesis, have specific markers, which you can then, essentially, if you want to characterize your preparations, you can see whether these markers are there. And then, of course, in these preparations, you need some kind of a negative control because lots of vesicles are produced by cells, and what we often use are these ER marker proteins like calnexin or calreticulin. So you want these ones to be present and these ones not to be present. Okay, so these are our cells again, the MDA-MB231 cells. We have the wild-type cells with caveolin, the short hairpin control cells with the same level of caveolin, and then the knockdown cells, which essentially lack caveolin. And then you essentially grow these cells, and you purify exosomes from the supernatant. And this is the scheme we use. Now, these cells, despite being highly metastatic, don't really produce a tremendous amount of exosomes. And so we first tried with these different ultracentrifugation protocols, didn't get very high yields, and ended up doing a combination of these centrifugation protocols with this exospin protocol to isolate the exosomes. Okay, and then, so we do the characterization here, which is basically looking at these vesicle populations by TEM, transmission electron microscopy, and what you can see here is in all cases we have actually vesicles. They're of roughly the same size as you can see in these images. And then you can also do NTA analysis, for instance, by nanosite. And essentially what that allows you to do is to determine, one, the size of the vesicles you isolated, and also the concentration. And so what you can see here is that the average size of these vesicles is similar in all cases. So this would suggest that we're getting similar types of vesicle populations from the different cells, and also the yield was reasonably similar. So it would seem that the presence or absence of caveolin did not necessarily affect essentially production of the exosomes. But what you can also see is, so this is the exosome peak here, but you can also see that there are other peaks, which means that we're dealing here with an enriched preparation of exosomes, but they're not only exosomes. So again, the characterization now. So these are the cells, showed you before, caveolin, caveolin, no caveolin, and here are the exosomes, caveolin, caveolin, no caveolin. So the caveolin that is present in these cells, to some extent, is also accumulated in these exosome preparations. Controls like actin are not present. We have marker proteins that are present in the exosome preparations here. But then, for instance, calnexin, the ER marker, is not present or to a highly reduced level. So then, now to an experiment. So one thing is to have a protein there or not, and the other thing is to get a function. And so the first thing we did is this type of experiment. So essentially take the exosome preparations we have and then treat cells and see whether we can get some type of phenotypic change or functional change. So we isolated these exosomes and essentially incubated then the cells with these exosome preparations. And we did, for instance, either migration or invasion assays. Here we're just looking at essentially invasion assays. And now this is treating the same MDA-MB231 cells with their exosomes. You would not necessarily expect an increase because you say, well, they already produce exosomes, but you actually do get an increase. So to treat them with wild type and then with the short hairpin control, you actually get significant increases here. And the interesting thing is that when you treat with the knockdown for caveolin exosomes, you actually suppress migration invasion shown specifically here. So this is quite interesting in terms of what the biology might be behind this observation. This we also applied to these cells that are called T47D cells, that are breast cancer cell line with low metastatic potential, do not express caveolin, and we get something similar here too. We get an increase in migration or invasion, not seen with the knockdown caveolin exosomes. So let's go to in vivo experiments. So this was all in vitro, all nice and good. Does this somehow translate into something that will be relevant in vivo? And for that, we changed our experimental model and used these BALB/C nod skid mice. And these are experiments that we did in collaboration with Lorena Lobos. She was instrumental in implementing these experiments. And so, again, what you do here is you isolate the exosomes, and then essentially first inject the cells, and then periodically the exosomes. Essentially 4 times, 4, 8, 12, and 16 days, and then sacrifice the mice at day 28. And we're going to look at results from 3 types of assays here. Cells in acidic fluid, tumor formation in spleen and pancreas, and then in the mesentery. So first, ascites, or accumulation of cells in ascites. So this is an indication of an extremely enhanced metastatic capacity of these cells. And what you can see is that the cells, the MDA-MB231 cells, treated with exosomes, either wild type or short hairpin control, have essentially a great capacity to accumulate in the acidic fluid, which the other ones do not have. And interestingly, in the case of the inoculation with the short hairpin caveolin exosomes, you don't get that. So it's definitely dependent on the presence of caveolin-1. If we look now at tumor formation in spleen and pancreas, the MDA-MB231 cells are able to essentially colonize these organs, and the exosomes treatment don't necessarily increase that, but if we use the exosomes without caveolin, we actually inhibit this ability again. If we look at now tumor formation or accumulation of tumor mass in the mesentery, you can see here that the MDA-MB231 cells obviously accumulate there, form tumor mass, and this increases when we incubate or inoculate the mice additionally with these exosomes containing caveolin. But if we use again these exosomes where caveolin was knocked down in the cells that produce the caveolin, they have an inhibitory effect. So essentially what we can conclude here is that exosomes with caveolin derived from highly metastatic cells can transfer this ability or metastatic potential, let's say, to less metastatic cells. And if you generate exosomes from these same cells, but after knocking down the caveolin, you generate the same amount of vesicles, but they not only do not have this ability, they can actually suppress the innate ability of the original cells. So there's a lot of interesting biology behind all of this. So this part is true. The exosomes with caveolin important, but then we did some experiments to look at, for instance, uptake of these exosomes. We saw that they're taken up in recipient cells to the same extent. And what we looked for was accumulation of caveolin in these recipient cells, and we didn't see that. So that suggests that it's probably not this mechanism. It has to be some other mechanism. And so just briefly, what we've done to try and figure out what might be happening here, thanks to collaboration with Carlos, sort of picture. It's a lot better than the photo you have of me. So this America did in Carlos's lab at UQ, basically run these gels, isolate these slices from the gels, run them through a preparation procedure, and then extract peptides and run them over a mass spec. And of course, you get a huge number of information, or a huge amount of information, looking at the exosomes from short hairpin caveolin cells, so knockdown caveolin, comparing them with wild type and short hairpin control. And we'll just focus on these 7 proteins here, because essentially they are the difference between these preparations, or the major difference, and this one here. So these are the ones that are only present when the exosomes contain caveolin. And they turn out to be these 7 proteins here. And the interesting thing is that many, or basically all of these 7, or 5 of them at least, have something to do with either the cytoskeleton or adhesion of cells, except this one here. And then so what we wanted to do is, of course, validate our proteomics analysis. Just because you find a peptide by proteomics doesn't mean you're necessarily going to find the protein. And essentially we were able to detect, we focused here on 3 of the proteins I mentioned, so tenascin, SIR61, and S100A9, just because we thought they were interesting, at least interesting to us, and essentially we were able to validate the fact that they're present in exosomes where caveolin is present, and absent or present to a reduced extent in exosomes where caveolin has been knocked down. Other proteins don't change, like these ones here, and these are the negative controls. So our little summary model here. Exosomes from these cells containing caveolin seem to contain these proteins that I already mentioned. And what we think, many of these have to do with adhesion in cells. So what we think is happening is that this is then transferred to recipient cells. They have an increased adhesive capacity, and also, as a consequence, then we'd have increased CSARC signaling downstream, for instance, integrin signaling. And this would contribute to invasion and metastasis. We validated our proteomics analysis for these three proteins. And of course, in the short hairpin control caveolin cells, these are essentially not present or present to a reduced extent. And so this would result in less activation of SARC kinase in the recipient cells, less invasion, less metastasis. This, however, is still rather speculative. So that's the same thing again. Caveolin-1 expressing metastatic cells produce exosomes with caveolin. They can convert a relatively benign tumor cell into a highly invasive malignant cell, and they do so in a caveolin-dependent manner. And the caveolin-containing exosomes, or EVs, are basically loaded with specific cell adhesion molecules. Just like to thank here all our collaborators, specifically Manuel Varas. He was really helpful with the analysis and characterization of our exosome preparations and in giving advice. Our international collaborators, particularly here, Carlos Salomon, the mass spec characterization of exosomes, funding, of course, these two centers, now funding by FondUp and Fondesit. Thanks. Also to Carlos.
That is fantastic. Thank you for the photo, by the way. Questions?
I have a question. I noticed that S100 was one of the proteins that you found. Now that's also a highly inflammatory protein and increased in a lot of inflammatory states. So again, demonstrating the link between.
Right. Well, it's a calcium binding protein, right?
Yeah.
So, which regulates a whole lot of things within the cell, yeah. No, I mean, there's a strong connection between caveolin and inflammation. In systems like, for instance, in fibroblasts, it's anti-inflammatory. But in other systems, it's, for instance, in lymphocyte, dendritic cells and things like that, its expression is induced by inflammation. So, and we have a paper with Alvaro Yatza where we show that in dendritic cells, for instance, pro-inflammatory cytokines will increase the expression of caveolin, and it's actually useful to the dendritic cells, so it aids them in directional migration then to the lymph nodes.
It's very interesting the effect of your exosome with the caveolin, that they can promote metastasis in vivo, compared with the exosome from when you knock down the caveolin. No? Did you try to incubate both at the same time to check if the exosome coming from these knockdown caveolin cells can abolish the effect?
No, whether it's dominant. No, we haven't tried that experiment, but it'd be interesting, definitely. Yeah.
Any other comment, question?
Nice talk, Andrew. You know, when I hearing your presentation, you know, we from our work, hypertension, we also study RAC1.
Okay.
Because RAC1 is induced the disease a subtype of hypertension by itself. Just RAC1 in a transgenic mice increase the reabsorption of sodium and water in the renal tissue. And also we found the Chilean population have two important polymorphisms in RAC1. And we studied that. And also we studied expression in monocyte in the population. So I was very impressive because RAC1 is also doing a job in, I don't know, cancer or other disease that we don't know.
Right.
I can show you the...
Sure, no, it'd be interesting.
It's actually public.
Right. So what kind of monocytes are these? Are these macrophages or?
Yeah, it's monocyte from the PBMC.
Yeah. Well, macrophages are loaded with caveolin, so. Yeah.
Well, probably final comment. Did you focus only the proteomics, so in the protein content of this exosome? But have you considered to check out other molecules that can be in that exosome, especially the microRNA that can be effective for this knockdown, the caveolin?
Next step. We're working on that. So the blue people in the group
Yep.
that I showed at the beginning, they're the ones that will be working on that kind of question, yeah.
Yeah, because, well, of course, you need to perform experiment, but probably the easy experiment to perform in using a couple of bioinformatics tools will be check if your protein that changed can be targeted from some of the microRNA that can be there. It's a couple of two websites in which you can see all the microRNA that have been published that have been associated with exosome already, and check if the microRNA that potentially can be targeting your protein of interest can be associated with exosome as well.
Oh, yeah, okay. See, yeah, great idea.
Okay, well, thank you, Andrew. That was fantastic.
Well, thank you.
So we have a long break now for lunch, and see you at 2:30, right, Felipe? Yeah, 2:30 here again. Thank you.